So I have the pleasure for introducing our last two speakers who are both members of the Department of Internal Medicine, and I promise to keep it short so I can't accuse them of going over time. So, so first is Frankie Heyward, who's been a member of the Center of Hypothalamic Research in the department for just under 1 year. A little bit about Frankie. He did his undergrad work at University of Delaware and then did a PhD at University of Alabama, Birmingham, where he studied the effects of obesity in making epigenetic changes in the hippocampus as it relates to learning and memory, and had a couple of really highly cited papers from his thesis work. He then went to Boston to work, a place I know fairly well, the endocrine division at Beth Israel Deaconess Medical Center and Harvard Medical School, where I worked with Evan Rosen and Brad Lowell, and did the first transcriptomic analysis of very important neurons that you'll hear about, HRP neurons that regulate food intake and body weight. So we're really excited to have Frankie in the center. Outside of his active scientific career, he's also involved in community outreach. He impressively is the founding director of the national and chair of the National Black Postdoc Association and the Harvard Medical School Black Postdoc Association. So Frankie, I didn't take too much of your time, so tell us about your work.
Joel, thank you for the really generous introduction. I realize I'm the penultimate talk. I commend you all for still staying here. So I'm going to be brief. All right, so my lab studies the epigenetic regulation of a neuronal form of cellular memory referred to as synaptic plasticity, and its impact in neural circuits that control energy homeostasis and in particular appetite. Our focus is on the hypothalamus, wherein lies a well-characterized melanocortin circuit that controls hunger and satiety. AGRP neurons represent a critical node of this circuit. Their activation is thought to be the cellular correlate of hunger, and they're often referred to as hunger neurons. The hormone leptin is a secreted adipokine that binds to its receptor throughout the body, the lion's share of which are in the hypothalamus. During the development of obesity, you get an expansion of adipose tissue and further secretion of leptin into circulation. And it has actions in the hypothalamus that put into play various physiological changes, such that you have an increase in energy expenditure and a reduction in food intake. And this helps shift the energy balance scale away from excess weight gain. Okay, so we have mice that are fed here. We have mice that are fasted overnight and then given vehicle, or mice that are fasted overnight and then given leptin. In the fed state, AGRP neurons are relatively quiescent. In response to fasting, they have a marked increase in their firing rate. And this is associated with changes in cellular changes in AGRP neurons that allow them to be more easily excited by upstream neurons. This is referred to as synaptic plasticity. They also have an increase in their intrinsic excitability as well. Furthermore, leptin suppresses this fasted-induced increase in AGRP neuron synaptic plasticity and intrinsic excitability. It should be said that alterations in synaptic plasticity and intrinsic excitability necessitate the dynamic regulation of gene expression by way of the actions of transcription factors. And so we're really interested in better understanding the manner by which transcription is regulated in not only AGRP neurons, but other neuronal populations that comprise the melanocortin circuit. This has been difficult to grapple with for two reasons. First has to do with the exceptionally high degree of cellular heterogeneity that exists within the hypothalamus as a whole, coupled with the fact that there are relatively few of not only AGRP neurons, but other neuronal populations in the circuits that we're interested in. So during my postdoc work, I made use of an engineered mouse line that built off of a line that Joel Elmquist generated. These are AGRP IRES Cree mice, in conjunction with a Cree-dependent nuclear and ribosomal tagging mouse line that my postdoc mentor Evan Rosen generated, referred to as NuTrap. And so I generated these NuTrap AGRP mice and was able to, in these mice that were either fed, fasted, or leptin treated, generate AGRP neuron-specific RNA-seq profiles. I was also able to sort AGRP neuronal nuclei and generate, using a TAC-seq, a readout of accessible areas of chromatin where transcription factors bind to either enhancer elements or promoter regions of genes and drive gene expression. I then computationally integrated these two data sets to infer the identity of transcription factors that are active and on board during states of fasted-induced hunger or leptin-induced hunger suppression, and populated a list of 100 or so transcription factor motifs that are implicated as being potential mediators of the acute hunger suppressing effects of leptin. One of the foremost enriched transcription factor motifs was that of interferon regulatory factor 3, or IRF3. IRF3 is a core component of the innate immune antiviral response pathway, and it hadn't been studied in this context previously. I then engaged in a battery of in vitro and in vivo gain of function and loss of function experiments to test a model whereby leptin works through IRF3 and AGRP neurons to confer its acute hunger suppressing effects, and substantiated that model. And then so what we think is happening here is as leptin levels fluctuate and go down in response to fasting and then rebound when you give exogenous leptin, so too does IR3 activity in AGRP neurons. I was also interested in identifying the direct transcriptional targets of leptin-activated IR3, and accomplished this using cut and run for IR3. And this allows us to appreciate direct DNA binding events of a constitutively active form of IR3 in AGRP neurons. And what I observed was that IR3 has the capacity to bind within accessible areas of chromatin that express the IR3 motif near genes that go up in response to leptin. I then tested some hypotheses. And so I suspected that leptin would work through IR3 to suppress hunger. If you had a loss of function of IR3, I predicted that we would see increased hunger and increased body weight. Conversely, if we had a gain of function of IR3, I predicted that we would see decreased hunger and decreased body weight. I actually did not substantiate that observation and saw the opposite, which was pretty perplexing. But that observation, coupled with an appreciation for a very provocative model put forward by Phil Scher, whereby hyperleptinemia that develops during the course of obesity is not only pathogenic, but obesogenic, allowed me to put together a conceptual framework that I'll describe to you. And so what I think is happening is in the acute setting, when you administer leptin, that's activating IR3 and prompting it to drive a transcriptional program that suppresses hunger. But during the development of obesity, where you have hyperleptinemia, IR3 is chronically or predominantly activated, and it's becoming deranged or functioning in a deranged manner and driving a transcriptional program that promotes hunger. And so I tested this model first by mimicking this chronic IR3 activation state in all leptin receptor expressing cell types. And when I did this, mice became profoundly obese. This is on a chow diet. And then when mice were only allowed to consume as much food as their control counterparts, it completely abrogated this increase in body weight gain, suggesting that it's driven by hyperphagia or hunger. Furthermore, I tested the model by deleting IR3 and placing mice on a high-fat diet. And in doing so, saw that these mice were protected from diet-induced obesity. And so I'm still working to elucidate the role of IR3 during the development of obesity, and this work is supported by my KO1 currently. So in my group, there are three big questions that we're after right now. So the first is, are there alterations in the neuroepigenome during obesity? And to accomplish this or attack this, we're obtaining human postmortem brain tissue from individuals that are not obese versus those that are, and comparing the neuroepigenome between the two. We're also continuing our work in mice that are either lean or subjected to diet-induced obesity and doing comparisons. Secondly, we're asking, does the neuroepigenome encode body weight set point? And so there's this really fascinating phenomena whereby individuals lose weight, but they have a tendency to regain that weight. And so I've mimicked this in mice that have been placed on a high-fat diet for a prolonged period of time, and then these mice are calorically restricted until their weight matched their control counterparts. And then I only allow these mice to consume as much food as control mice for either 0 days, a week, or a month. And in all cases, when the mice are allowed to eat freely or ad libitum, they regain a substantial amount of weight back. So what we think is happening here is that body weight set point at some point is being shifted upward. And the question we're after is, how might this body weight set point be encoded? And could it be encoded at the level of the neuroepigenome in these neuronal populations that control energy homeostasis and appetite? And so if the answer is yes, I mean, can we detect this using some of the approaches that I'll describe? And if so, can we reprogram the neuroepigenome and in turn reprogram body weight set point and prevent this weight regain from occurring? And then lastly, I'm asking, is there a neuroepigenetic basis for reduced hunger during aging? So there's clinical evidence that individuals, as they age, exhibit reduced appetite. This has been seen in humans and in mouse models as well. And we're doing comparisons between young and aged individuals while comparing their neuroepigenomes. In much of my work, I'll be employing CRISPR activation and inhibition approaches to, with a great deal of precision, reprogram the neuroepigenome. And lastly, we'll also be using single-cell multiomic approaches to generate single-cell RNA-seq and ATAC-seq data sets. For instance, I have the capacity to enrich for all leptin receptor expressing cell types. And I see around 39 different clusters in our RNA-seq and ATAC-seq data. We see really interesting populations like AGRP neurons, POMC expressing cell types, and then also populations of cells that express GLP-1 receptor, which are likely the targets of the blockbuster anti-obesity drugs, semaglutide, Ozempic. And importantly, across our different cell populations, we see distinct transcriptomes and chromatin accessibility profiles. And so with that, I'd like to thank Evan Rosen for allowing me to develop this research program in his group during my postdoc and instructorship, and thank my past and current collaborators, as well as my funding sources, and you for your attention.